Hi, I'm Christina Holder, and I'm a student at LaTourneau University. My research project is testing the reliability of DNABInd and DNAbinder software for identifying DNA binding proteins in bacteriophages. So the idea of my research was born earlier this semester while I was doing functional annotation for the phage dynamo that I had isolated last fall. So as I was looking at functions in a certain operon, I noticed that most of the genes I was identifying were either DNA binding proteins or hypothetical proteins that I was not able to find a function for. So out of curiosity, I decided to run all of those hypothetical proteins through DNAbind and DNAbinder. Interestingly, I found that most of these proteins had very strong probabilities to be DNA binding. So although I was not able to call any of these functions since there were no strong NCBI BLAST or HH-pred hits, this did bring up the question of if we could trust these softwares to make future calls for gene function. So to test this, I chose 6 different clusters of varying hosts and life cycles. And then from each cluster, I selected 3 random phages. And then using Famirator, I found 8 DNA binding protein sequences and 7 non-DNA binding protein sequences from each phage. I then ran all of these sequences through DNAbind and DNAbinder. So in the end, I ran a total of 270 sequences, of which 153 were DNA binding and 127 were non-DNA binding. So the data of the reliability of these softwares can be seen in these tables represented as sensitivity, specificity, positive predictive values, and negative predictive values. So a little background on each of these terms. Sensitivity is the percentage of DNA binding proteins correctly identified out of the total number of DNA binding proteins run. And then specificity is the percentage of non-DNA binding proteins correctly identified out of all the non-DNA binding proteins run. So we are looking for a high sensitivity, which would mean that the software is able to identify a large number of DNA binding proteins, and a high specificity, meaning the program does not call many false positives. So positive predictive value is a bit different. That number gives us an idea of the proportion of true positives called out of all of the DNA binding proteins run. And then negative predictive value shows the percentage of true negatives called out of all of the non-DNA binding proteins run. So the idea for this project was to create a combination of both of these softwares to use for functional annotation, similarly to how we use a combination of softwares while identifying transmembrane protein domains. So to identify the most reliable data set of DNAbinder, we can look at the sensitivity and specificity rates. So as you can see, the realistic data set has a very low sensitivity, meaning that it is not going to be able to call a high number of DNA binding proteins. So that means that that data set would likely not be very helpful. So that leaves the main and alternate data set. And as you can see, the main data set has a higher sensitivity and specificity than the alternate data set, meaning that out of all the DNAbinder data sets, main would be our most reliable. Moving on to DNAbind, as you can see, there are different rows for different false positive rates. So on the website, there is a false positive rate default of 15%, but this seemed a little bit high, so I wanted to run various lower false positive rates to see if I could get a specificity of 100%. And as you can see, that was successful in that the false positive rates 1, 5, and 7% gave a specificity of 100%. So 7% false positive rate still sounds relatively high. So I would recommend that we use the false positive rate 1% or 5% to complement with the DNAbinder dataset main. So in the future, I would like to expand my data set, including more clusters and more phages. And I would also like to do some wet lab experimentation to get a more accurate sensitivity and specificity rates for these softwares, specifically on hypothetical proteins. And I would also like to look into developing a DNA binding prediction software specifically for bacteriophages. So in conclusion, I think that using DNAbinder run on the main data set with a combination of DNAbind on a false positive rate of 1 or 5% could be very helpful for functional annotation of hypothetical proteins. Thank you so much for listening. I hope you enjoyed.